I got this book on biochemistry. It's called Biochemistry, A Brief Course, written by Mazur and Harrow. Here's the front of the page, or the front cover. And the copyright date on this book is December 1969. I guarantee you a lot of this stuff is still applicable. And what I wanted to talk about really quickly is, since we all understand, all my audience understands, that the fusion model of stars is absolutely false, that elements are synthesized during galaxy birth, where the jets come out at almost luminal velocities, where that's where the elements are synthesized. But inside of stars are the places where biochemical, electrochemical reactions take place, redox reactions, that kind of stuff happens. So, in essence, as the star evolves, it forms life. And some of the basic building blocks of life were shown to be possible via the Miller-Urey experiments, and which produced more than 20 amino acids. But the 20 amino acids that we're familiar with, it says here, classification of amino acids on page 19. I'll just go ahead and name a few off. Glycine, alanine, valine, leucine, isoleucine, phenylalanine, triosine, tryptophan, serine, threonine, aspartic acid, glutamic acid, asparagine, glutamine, lysine, did I already say that one? Leucine and lysine, arginine, histidine. I can't even pronounce these. But it has them all written out. You can see their chemical formulas and all that good stuff. And, it actually doesn't go over their structure. I mean, I'm sure they probably look different if you were to draw their structure out, in those diagrams. But those are the chemicals that are inside, hydrogen, nitrogen, carbon, oxygen, that good stuff. And that basically means that the interiors of extremely evolved stars, via the stellar metamorphosis theory, contain amino acids, because that's where life begins. It's inside of a stellar interior, a really, really evolved star. And those amino acids are synthesized via very wide-ranging chemical exothermic reactions and various other reactions. And one of the really cool parts that sticks out are the properties of amino acids and the polypeptides. Peptide is I think that's the amino acid when it's connected to another one, I believe. I have to do some more research on it. But polypeptides meaning many, poly meaning many, and then peptides, many amino acids. So, as here, the synthesis of amino acids, you have your amination of halogenated acids. There's a malonic ester method, the Stricker synthesis, and the synthesis of polypeptides, which are acyl chloride method. Let's see, it just has a lot of good stuff on there to study. And you have carbobenzoxy method, or the Bergman synthesis. And you have, let's see, what else do you have here? Polypeptide synthesis in the solid state, Merrifield. It's pretty interesting. I have to do a lot of research and study these to the best of my ability, because this is what's occurring inside of Neptune and Uranus as we speak. They're not these ice cold worlds where nothing interesting is happening. They're pre-Earth, if you will. Well, all of the stars are pre-Earth, but those are in the very later stages of stellar evolution. They're going to become ocean worlds, and obviously, as also in here, you can even classify these amino acids based off of their solubility inside of solutions which contain a little bit of salt and different properties. You can sort them according to the solubility. You have your albumins, your globulins, your gluteins, your gliadins, or prolamines, your histones, and protamines. It also has a list of all of them right there. I don't know if you can see that. And you can classify the amino acids also based on their structure. You have your fibrous proteins, your globular proteins, and there are lots of different ways you can classify them. And depending on how you can classify them, you can also determine where it is that they might be evolving as the star's interior evolves, because my audience also knows that inside the star, that's where it forms the oceans, the water oceans, because the hydrogen and the oxygen combine together, creating vast oceans. And then depending on how much water is there and how salty it is, or if it has a lot of methane in it, or, you know, depending on how pure that water is, will determine which kind of amino acids will be able to be synthesized at that level inside the stellar interior. So you have to, you know, go over that as well. I think the whole idea of having life form on meteorites and then brought to Earth, I don't think that's reasonable. Also, another thing I wanted to point out is that all of these molecules are comprised of oxygen, hydrogen, nitrogen, carbon, meaning it happens towards the very top portions of the star. So the heavier molecules in the bottom and the top portions. And some of the amino acids contain sulfur. So my best bet is that those amino acids were forming in the lower regions, much closer to the surface of the very, very deep interior of the star, where a lot of the other material was, like magnesium, iron, nickel, and other heavier elements. So, you know, you can look at the amino acid, and just like a rock, where you can look at a rock and say, oh, well, this is granite, so it had to be up here, or this is like gneiss, and it formed really, really lower, a lot higher pressures than granite did, even though it's the same composition. That must have formed lower because of, you know, of the folding inside of the rock. You can do the same with amino acids. Like, you can look at the amino acid and determining what kind of elements are, you know, inside there, and, you know, if they have triple bonds inside of them and stuff like that, and you can determine where the amino acid was probably synthesized inside of the stellar interior based off of its structure and of its composition. Of course, some amino acids probably have stronger structures than others. You know, just like when you have diamond and you have pencil carbon. The pencil carbon is really smooth, and it's, you know, it can fall apart really easily, but diamond can withstand a lot of pressure. So you could probably classify a lot of these amino acids based off of their ability to be stable under high pressures and temperatures. But yeah, that's that. I have a lot of reading to do. This is a very, very, very good book, very high quality, and you know, has lots of good diagrams in here and things, and goes over nucleic acids, goes over let's see, what else does it go over? It goes over enzymes. I want to go into enzymes and catalysts. I also want to talk about platinum, how platinum is a very, very, very small catalyst and could probably be one of the very first enzymes for life formation. I haven't seen anything like that written anywhere, so there's probably something there as well. But yeah, I have a lot of learning to do with this, and hopefully, you know, photosynthesis, you know, lipid metabolism, lipids, carbohydrate metabolism, things like that. There's a lot of stuff in here I have to review. Oh, here's a summary of various aspects of the Kreb cycle. That looks pretty interesting. But there's a lot of stuff here. I mean, this is way more than what I learned in my basic chemistry and biology classes in college. So hopefully people can really get the point, is that when the star evolves, not only does its physical structure evolve, but life evolves inside the star as it cools and dies as well. That's the main point I want to get across. It's not a fusion reactor at all. Fusion happens inside of galaxy birth, where the energies are vastly higher and much more violent than anything that could ever happen inside of a star. Alright, later. Oh wait, today is October 4th, 2015.